you have to do or what you should do while doing the meta-analysis. So first I want to start off why in general you do not want to do a meta-analysis, and I think you know that, but I just reiterate. First, you can base your inference on huge sample sizes, so different studies, they in a way come together in the effects you see. Then they identify effects that are reproducible over studies. Then you can integrate variants of probing a cognition, as we heard just before, in behavioral domains or in paradigms, over paradigms. And we can relate topics or paradigms that you can't really study in one only study. So a meta-analysis allow you for a lot more than single studies. But it's different in a way as well. There's a very good cookbook just now. I want to differentiate between different meta-analysis. So it's the classical thing to do meta-analysis would be, or an image-based meta-analysis in contrast to coordinate-based we just heard, which would be a mega-analysis when we have all the individual maps, unthresholded T-maps that we would combine in a mega-analysis. Then we would have effect size good and really could look at that in the best possible way. Multi-study conjunction would be another way of going to the actual raw data and combining this by overlapping significant effects, for example, on the group level. And third level analysis would be between experiment effects, where we combine raw data as well. The problem there is the compilation of original data was not possible in the early days at all, really. And it's really difficult now to find enough original data or original studies to do a big image-based meta-analysis. But there are databases out there to make it easier. But what you can do using, as we just heard, our coordinate-based meta-analysis, based on published maximum coordinates. And these have sparse representations of the results, but they may allow the integration of the entire literature, really going back the 20, 30 years since MRI and PET started. Now, how you would do that, and what I'm present now is true for image-based analysis and coordinate-based meta-analysis. There's a very, very good paper out just now, which is 10 simple rules for neuroimaging meta-analysis, and you should read this. I mean, not should, you have to. If you want to do a meta-analysis, do that one. The point is, it's on a Creative Commons license, so anybody can just download it, and here is the link to that. The point is, it will give you very detailed and thorough view what to do, and what it gives you also is a flowchart. So what are the steps that you actually have to go through to do a meta-analysis, as well coordinate or imaging based, and it provides like it's like a questionnaire for yourself or a reporting scheme of what you do on the way. And when we're talking about reproducibility and replicability, we have to do this. So, and I'm pretty sure that all the people, all the reviewers will ask for this in the future. So do it early on. Now, I would go through the 10 simple rules, but I'm trying to go through I will go through the flowchart to have an idea what you have to do or what it means to do this. So the first steps are specification of research questions, then the literature search, data extraction, and very important, double check your data. So first, specification of research question. What do we mean by that? First is the definition of process of interest. I'll just take working memory here. So we were interested in that. And that's then looking at inclusion-exclusion criteria or paradigms and contrasts. You have to be specific about this because we know working memory can be probed by doing different tasks. Sternberg, Anbach, delayed matched sample are few, but there are many more. And you should not only look at all these, but report it, which ones you include. Those can further be applied in very different way. So stimuli that are verbal items, spatial locations, and so forth. You have to look at all these. It's not enough to just say we did working memory, collect the coordinates. Then, and you can investigate them even further. So the point here is it's getting very fine-grained in every paper, and you really should note that down and specify beforehand what you're interested in. You want all of these? Yes, but maybe you're interested in a few of these. Now, there are other inclusion-exclusion criteria. For example, if you go coordinate-based, that's the example I'm using here as well. You only include studies that have voxel-wise whole brain analysis statistically, because otherwise the coordinates representing something else in that sense. So it may be in inclusion-exclusion criteria. You can use ROI as well. Maybe this. No ROI analysis if you use classical coordinate-based analysis. No small volume correction, no partial brain coverage, because all this will be problematic using specific statistical corrections afterwards that you have to do in meta-analysis as well. Then the coordinates in which schemes, so are there in MNI space or Talaris space, note all this down and or be sure that you specified it in your inclusion and exclusion criteria. So these are parts of this, to know when you find actually something to put it in or not. Be careful, because as we heard, the legends or how they report isn't always right. Look at the coordinates where they are in the brain. And it's good to know that SPM, FSL, and SMNI space, but AFNI and brain voyager, for example, it depends of how it was used in the study. So don't believe everything you read there. I know it's general, but have a look yourself, right? On if it's probable, it's in the brain, outside the brain, so on and so forth. Okay, and if the coordinates are transformed, and be sure before you start what you want to include. Now there are other. This is just a general scheme. There are general selection criteria. What I just and specific selection criteria. General, what to include, yes or no, and specific. Do I want this paradigm, yes or no, and how it's described? But there's others that are exclusive. So you have normal mapping, where you look at cognitive functions. So basically, when I do working memory task against when I do a motor task, that will be a mapping of the cognitive function. And if I do patient mapping, what we heard as well, if you do a contrast between a patient group that does a working memory task or a control group, and you can't put them together because the coordinates represent really something different. So be sure about these selection criteria beforehand. Now, normal mapping, it means as well no interventions. So no pharmacological TMS or training, because then it's not the function per se, but the variability of this. Then include healthy subjects, so no patients, but you can use the patient group, in a patient study, the control group, if there's the effect. No selected groups, so no genetic specific groups or behavioral like experts, because those are expert studies. These contrasts can be difficult because it's not the general function. And in general mean between condition effects, an fMRI and PET in general. And on the other side, we have patient mapping like group comparisons on any level. That may be an fMRI contrast between patients and groups or between young and old. And on the other hand, like maybe a atrophy that is more prone, that's happening in Alzheimer's against like control groups. You can do it on VBM as well. So between groups effects or between condition effects. You can combine these, but they're separate analysis, okay? And define those as criteria before. Now we do, as we have criteria that we want to impose, we use systematic literature search, and there we can use databases for that search, keywords, and but be sure to use date boundaries. The point there is you can use BrainMap to have a look how many studies are there already in BrainMap. It's very well curated, and you have quite some studies already for a lot of functions. But you can, and there are others. There's Neuroscience, for example, another big database that another approach, and has a lot of studies in it. And BrainSpell is a new version that uses the data from Neuroscience and curates it. Lastly, there's another database, the ANIMA from UD, from us, where you can look at actually meta-analyses that were done, and actually can look at the maps, the statistical maps you can look at. So use these to have an idea, is there a meta-analysis done already? So is it worthwhile to go through? Or how many studies are there that I can build on? But then, additionally, you should do literature search. And that's like the classical thing to do when doing meta-analysis. Now we use working memory as a term. We put it in, but we put it in different ways. So working memory and fMRI and healthy subjects. Note this down, like really every single search you do. That's very important to replicate the study, together with date boundaries. When did you do this search? The same is true for databases because they evolve. But this is not enough. You got to search for synonyms, related topics. You use different search engines and the internet per se. And you use review articles, do reference tracing. One thing is look at one of the databases, look at the papers there, and then on the reference they use. But note it all down. That's really important, because otherwise you go back and have to do it again. Okay, now we're at the stage where we do data extraction. So we note down all the coordinates. We note down sample sizes, inference. So everything you find in your in the studies you found through your keywords, you have to note down to be used later for your analysis. So the point there is every information you can find in the articles, note it down, and you have to work on your scheme how to note it down. There's quite sometimes when you have to go back, okay, there's something specific contrast I didn't thought of. Do I want it as included in the study? So at this stage, you can go back and forth because you didn't do any analysis yet. You won't inflate any statistics, or you don't have to correct for any comparisons because you didn't do any. And based on how many you found, you may think about, okay, it does not make sense to use a specific contrast or to use specific group characteristic. So this is really a process that you have to go back and forth here. And there's just for what is needed for GPR, so it's Gaussian process randomization. I think it's a way of using images or is it called? But it's a way of doing statistics on meta-analysis, and the ES-SDM is a combination of coordinates and original like statistical maps for meta-analysis. So it's not one or the other. There are algorithms you can do both. And if you have studies and coordinates, you can combine them as well. Okay, now we come to, you note it down. This is just an example how you could do it. So note down the paper, note down how many subjects, what kind of contrast, what kind of task was invoked, modality, stimulus type, if it was fMRI or PET or VBM, and make comments to all of this. Because all of these comments, you will make new categories if it's important on the way. And I would even say note down the ages of the age range, for example, and so because it may be that certain functions are mainly looked at in elderly. And if you just put this in as the cognitive function in the brain, and most of the people are over 60, let's say, then it's a specific group where you find this function. So it's note down everything, and the reviewers, if it's not clear, will ask you anyway. So do it early on. Now, we have one study, as you heard before, a study and an experiment is not the same. One study, we would use the term for a paper. So a study is a paper, and any experiment in any paper there are classically in psychology where there were several experiments on different or the same subjects to actually show something about some cognitive functions. But here it's used there's every contrast, meaning task against control or task against baseline. These are different experiments, and for example for the Axmal study we have 4 experiments in one study, and use these terms right. So papers and so organize your data again. The type of contrast is really important because sometimes you want only a specific cognitive function. If you only use the baseline contrast, then you will have a lot of functions lumped together there. And if then it may be that you find an effect that is not specific for working memory in that sense, but for the task. So paradigm is important, contrast is important. Same like for the task. So if you have too many n-back tasks, only a few Sternberg, you will in the end do a meta-analysis on n-back tasks, not on working memory in general. So have an idea that it's balanced as well, or kind of balanced between tasks. And if you have enough studies, you can do a meta-analysis for Sternberg task, one for the n-back task, and even contrast to have an idea which one probes which part of the brain, which we heard before may even be interesting to see which one to use to get at a certain brain area in a patient population, for example. Then the stimuli type. I mean, if you have more studies, how you don't do the same study, the people won't do the same study again, so you won't get funded for it. So you have to change something, and one of the common things is to change the stimuli from the stimuli type, maybe letters or numbers or faces or tones, and be aware of that. So if it's all the modality is one thing, but the way how to present the stimuli is another. And we know that the general function of working memory will be there as well. But if all it's letters and there are a few shapes only in there, then you have like the cognitive function of semantics as well in there and of reading. So be sure that you know what are the main stimuli types and what others are used there. If there's variable enough, you can make the claims to general function. And we tend to overgeneralize. So here you can be sure of that. Now, going back to the specification of research question, we found enough studies. We are sure that we meet the inclusion and exclusion criteria, specification of paradigms and contrast. We have our table. Now we go through it. So which paradigm type is there? Because it determines then what is represented in the meta-analysis. Then the contrast types are there. So low-level baseline, or do we have against rest of fixation? Then we have a lot of different effects in there. So cognitive functions, or high-level contrast, where we have one specific cognitive effect of, say, short-term working memory. Further, there may be post-hoc classifications you can that may define with coordinates or effects. So how many were forgotten, how many were remembered, how high was the memory load, and so on and so forth. If you have enough studies, you can use all this to further probe which part of the area of the brain is associable with these things. So it really pays out in the end that you can enrich your meta-analysis and not only do one and do that. So, and the rank correlations, memory load, I mentioned before. Covariate correlations are another thing that's rather tricky. If you have reaction time correlations with your working memory, is that working memory? Not sure, right? It may be. So if I'm very fast, but it's an interplay. So basically, the reaction time, how fast I am, has something to do with memory, but the reaction time correlation may be another area depending on what kind of interference the task is doing. So basically, if there's an emotional distractor on working memory, I won't be as fast. And maybe the reaction time will be more associated with an area that's emotional related, basically. So you can use them, but be careful. And if you put them all together, you can't be specific about what actually is the brain, is the activation associated with afterwards. So that's and there's many more of these interactions. So this actually the specification of inclusion and exclusion criteria and so on and so forth determines what the results will represent. And this is really the main part for interpreting what you have at the end of the statistical valid inference you do at the end. Okay, now we did this well. We double-checked our data. Now we go further in having decisions for the specific analysis and potential sub-analysis based on how many studies we have, and we converted all the coordinates well, or the SPM maps if we have them in the same space. It's the same. So how many experiments do we need actually to do a sub-analysis? And for ALE, we did a large-scale simulation, and there we could say that around 20 experiments we can use the ALE algorithm to be rather sure, or to be sure that it's not driven by only one or two experiments, but for more. So we do an actual meta-analysis if we do cluster level correction with using ALE. The other thing, it doesn't hold directly for the other approaches. And it's not the thing that you can't do any meta-analysis with only 20 experiments. The point is you got to be careful of what you claim. If the threshold you apply, you have to be a little careful more to look at this. Main point is if there's not much variance of the coordinates and of the task, if it's all finger tapping, then and they're all only in a specific area, then you may need less experiments to have a meta-analytic result, but you have to report this. And if you do more analysis, you say it's exploratory, right? That you have to state this as well. Now, in the end, you calculate your meta-analysis based on what you did before. You get significant values, clusters, areas. And what is very important there, you have to transparently report what you did. And so all the things you did before, you hopefully all noted down. And then you can publish this in the best way, and pretty high as well. So the good thing is you have, for all of this, you have this really nice table. You can't see it very well, but it's in the paper. It's free. Download it and use it early on. Just note down the contrast you want to put in. Do it again, and if you store this away in a good way, just then submit it with your study, and you can be sure that you don't have a problem. And if you have too little, not enough experiments, and you noted it down well, and say, okay, it's exploratory. I got to be careful with the statistical correction I did, because I'm not sure, but I did it well. This is what I did, and it's interesting. It's still interesting if you have a small number of studies and you find something if you report it well. Now, these are things I think are important. This is the way to get these tables, and Thank you for your attention.